Hi guys, Miss Francis. I am here today to talk to you about the nucleus and the ribosome. The nucleus houses our genetic library, and by us I mean eukaryotic cells, and by genetic library I mean our DNA. Not only is DNA going to be found in the nucleus, but some genes are going to actually be found in the mitochondria and our chloroplasts. The nucleus is actually the largest organelle, and it's about 5 microns in diameter. The nucleus is separated from the cytoplasm. It's surrounded by a nuclear envelope, also known as the nuclear membrane, and it's made out of a phospholipid bilayer, hence double membrane. Within the double membrane, you have pores, which are holes. That's what allows macromolecules, particles, messenger RNA to travel through the nuclear membrane. The nuclear lamina is what lines the nuclear envelope, and it is going to maintain the shape of the nucleus. So here you can see the pores, and you can see embedded ribosomes that'll be found in the nuclear envelope. And then here you'll notice the nuclear lamina, which is just, what gives the nucleus some shape. So within the nucleus, like I said, it houses your genetic material. It houses your DNA. And because we're eukaryotic organisms, it's going to house DNA that has associated proteins known as histones. Chromatin is simply uncoiled chromosomes. When the cell begins to divide, though, the chromatin is going to coil, and now we call that chromosomes. And those are the structures that we're used to seeing in typical pictures of chromosomes. So humans, we have 46 chromosomes, whereas the egg cell and the sperm cell each have 23, which makes sense because they're going to come together to form 46. The nucleolus is what creates or synthesizes your ribosome. So in the nucleolus, ribosomal RNA or rRNA is synthesized and assembled with proteins to form ribosomal subunits. The ribosome itself kind of looks like a hamburger bun, and it's going to be composed of 2 subunits. And it's within this subunit that you're going to create your protein. So the nucleus directs protein synthesis, because within the nucleus you have your DNA, and it's the DNA that gives that code, that sends that message on how to make that protein with the messenger RNA. The process of the DNA giving its message to the messenger RNA is called transcription, and that's going to occur in the nucleus. When we do cell division, we'll get into the specifics on transcription. This slide is a nice summary slide regarding information on the nucleus, as well as this slide, which provides some more information. When they say some cells have multiple nuclei, those cells are the ones that are the exception to the cell theory. So here's a micrograph of your nucleus, the nucleolus, and chromatin that's going to be found inside the nucleus. Surrounding the nucleus, you would have your nuclear envelope. Ribosomes build the cell's proteins, and we all know that without proteins, we wouldn't be able to sustain life. The ribosome is composed of RNA and proteins, where you're going to have a large subunit and a small subunit. So the messenger RNA is going to thread its code in between the 2 subunits, and transfer RNA is going to bring over the appropriate amino acid. The amino acids will then be linked in their specific sequence, which is ultimately dictated by the DNA. Some examples of cells that would have lots of ribosomes because they make a lot of proteins would be your pancreatic cells. The pancreas makes insulin. It's also going to make glucagon, which are both hormones. Hormones are proteins. So the pancreas is going to have a large number of ribosomes. Your plasma B lymphocyte, which produces antibodies, which are proteins, are going to have a lot of ribosomes. Now, there are two different types of ribosomes. You have free ribosomes. You also have bound ribosomes. The key differences are that free ribosomes are suspended in the cytosol. Bound ribosomes are attached to the rough endoplasmic reticulum. Your free ribosomes are found in prokaryotes, whereas in eukaryotes, you have both free and bound ribosomes. The key difference is what types of proteins are made by the free ribosomes and the bound ribosomes. So the free ribosomes are going to synthesize proteins that function within the cytosol. For example, proteins that are needed within the cell. Whereas your bound ribosomes are going to synthesize proteins that are either included into membranes or needed for export from the cell. So that would be like your hormones, like insulin. So free ribosomes, proteins needed within the cell, such as digestive enzymes like catalase. Bound ribosomes are going to produce proteins needed outside the cell, such as insulin. Here's a nice micrograph of free versus bound ribosomes. So you can see that the free ribosomes appear as dark granules in the cytoplasm, and once again they synthesize proteins to function in the cytoplasm for use within the cell. And just to wrap up and remind us about the organelles that are parts of the endomembrane system, let's revisit that. So the organelles that are part of the endomembrane system include the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, vacuoles, and the plasma membrane. Next time, we are going to investigate a theory known as the endosymbiotic theory. And from there, we're going to get into specifics regarding the organelles that are part of the endomembrane system.